Imagine you've got a treasure map, but it's so small and faint that it's nearly impossible to read. Wouldn't it be great if you had a magic machine that could create hundreds of clearer copies? That's a bit like how PCR, or polymerase chain reaction, works with DNA. PCR is a special laboratory technique that helps us make millions of copies of a specific DNA sequence from just a tiny starting sample. So, how does PCR actually work? The process is fascinating and involves a few magical sounding steps that repeat over and over. First, we have the denaturation step. Here, the DNA is heated to a high temperature, causing the double helix to split into two single strands. It's like unzipping a coat when you enter a warm room. Next up is annealing. Imagine tiny helpers called primers who are eager to find their spots on these single strands. These primers are short pieces of DNA that attach themselves to the DNA we're interested in copying. Once attached, they wait for the next step. Here comes the extension phase, which is almost like building new roads. An enzyme called Taq polymerase comes along and starts adding new bricks or nucleotides to the primers. It helps create new DNA strands based on the original pattern. After these steps, the magic moment of amplification happens. The machine repeats all the steps again and again, doubling the DNA every time like a photocopier working in overdrive. But why is PCR so important? It's used in so many different fields. In genetic testing, it helps doctors find genetic disorders. It's used in forensics to analyze crime scene DNA, and scientists use it in research to study genes. There are different types of PCR too. Standard PCR is the classic method, while real-time PCR measures and tracks DNA amplification as it happens. Then there's reverse transcription PCR, which deals with amplifying RNA by turning it into DNA first. Of course, PCR isn't without challenges. It's so sensitive that contamination can lead to mistakes. Designing the little primers takes care and precision, or else they might not work correctly. In the end, PCR is like a wondrous laboratory tool that unlocks the secrets of DNA. It allows scientists to explore the fascinating world of genetics and makes our understanding of life's blueprint clearer than ever before.